Okay, this experiment is just a little bit more complicated. So let's start with this. This is Heredity and Evolution Project number 2. In part 1 of the coin toss experiment, we saw how the predictions made for flipping a coin was related to the predictions of inheritance from using a Punnett square. In part 2 of the coin toss experiment, we're going to practice coin flipping and Punnett square making. So the instructions on this experiment are to flip 2 coins at the same time 50 times. All right, so I flipped the coins 50 times, and if I landed on heads, it was a capital. If I landed on tails, it was a lowercase. So I recorded my results. I got 10, 33, and 7. I divided each of those out of my total number, which was 50, to find the percentages. Then I took my 2 highest percentages. I chose those as the parents. Okay, and in my case, it was homozygous dominant, capital B, and heterozygous, which is capital B, lowercase b. From there, I plugged those in and made my Punnett square. Okay, so those are the first steps of this assignment. The next steps of this assignment are to answer the lab write-up questions. So for question 1, you're going to describe in your own words the steps you took for filling out the Punnett square. And you can use this Punnett square in your description. Please make sure you're using scientific vocabulary and you're writing in complete sentences. For step 2, I plugged in my information here so that you could see, according to these results, what my information would be. So parent A genotype, or for A, parent 1 genotype and parent 2 genotype would be homozygous dominant, capital B, and heterozygous, capital B, lowercase b. For part B, the percent chance for either of those is 50-50. So you can see from my Punnett square, it's 2 out of 4 being homozygous dominant, or 2 out of 4 being heterozygous. So that's 50-50. 0% chance on this one of being homozygous recessive. E, for the genotypic ratio, that would be a 50-50 shot of having either homozygous dominant or heterozygous. For F, the percent of an offspring having the dominant trait is a 100% chance. The percent of them having the recessive trait is a 0% chance. And the phenotypic ratio would be 100 to 0. It's just 100%. Okay. The next part on our lab write-up questions: how close were the results of the coin flip to the genotypic ratio from your Punnett square? So look up at your coin flip results and see how close those numbers were to your genotypic, and then for number 4 to your phenotypic ratio. So mine, for my genotypic ratio, was 50-50. Those were pretty far off because mine was about 60-20% on the genotypic ratio. For the phenotypic ratio, it was a lot further off because I had 100% chance, and here we had when you add those two together, about an 86% chance. So that's just changed those a little bit. What could be done with the coin toss to make the results match the Punnett square better? Well, we could flip the coin more often or more times to get better data and do this more frequent so that we could have more samples. And we could also use varied coins. So we could use the same coins, or we could change up the coins and see if that has a difference. Okay?